Today's lecture focuses on the determination of sex and chromosomes that are involved in sex determination. But before we jump in, I need to include a few definitions just to make sure we have a good understanding of what we'll be discussing. And the first is a basic biological definition of sex. This is a classification of many, if not most, organisms based on their reproductive organs, and this is usually dictated by chromosome composition. This is in contrast, however, to common terms utilized in today's society referring to gender. Gender is defined as what an individual identifies oneself as with respect to sex. So, in this particular lecture, we won't be focusing on gender, which is an individual's identification of oneself. We will, however, focus specifically on sex and the chromosomal composition that determines the sex of an individual. Additionally, we have to discuss a few more definitions, and that is to understand or get a review of life cycles and reproductive modes. This is something that you should have covered in previous courses, so this should just act as a little reminder so that, again, we're all on the same page as we jump into the lecture material. The first definition, or reproductive mode, deals with asexual reproduction. This is a formation of a new individual from cells of a single parent. In asexual reproduction, there is no fertilization of gametes that takes place. Typically, in asexual reproduction, this occurs via mitosis, and what happens as a result of asexual reproduction is that this produces genetically identical individuals. There are plenty of examples in nature of asexual reproduction. Bacteria, for example, reproduce asexually most of the time. There are other examples, however, in the plant kingdom. There are examples such as stolons. Stolons are above ground runners that can form new roots and hence form a new plant. Strawberries are known to do this and reproduce in this way. The new plant that is produced from a stolon or this above ground runner is genetically identical to the individual plant that the runner came from. Tubers are another example in the plant kingdom that are components or the result of asexual reproduction. Tubers are underground storage stems from which new plants can grow after a dormant season. Potatoes are a classic example of this. And again, in this situation, these are individuals that are produced asexually, no fertilization of gametes is taking place, and you have genetically identical individuals that are being produced. There are examples of asexual reproduction in the animal kingdom as well. This typically occurs via a process known as budding. Jellyfish and starfish are known for this particular mechanism. In fact, you may have seen pictures where a starfish has lost one of its arms, and that arm then develops into a new individual. This again is a mechanism of asexual reproduction. Sexual reproduction, on the other hand, is reproduction through the fusion of gametes. This is how most diploid eukaryotes reproduce. And most importantly, with sexual reproduction, the key here biologically is that there's a mixing of genetic material, which then results in the diversity that we have discussed in the previous lectures. Lastly, one additional mode of reproduction is known as the alternation of generations. In this particular situation, there is an alternation of forms or modes of reproduction in the life cycle of an organism. Oftentimes, this is switching between a diploid and a haploid state or sexual and asexual reproductive cycles. But there are examples that exist of alternation of generations in nature. And lastly, some additional terms that we need to be aware of. That is primary versus secondary sexual differentiation. Primary sexual differentiation refers to the gonads, specifically the structure of the reproductive organs. Secondary sexual differentiation involves the overall appearance of an organism. Sometimes this is due to chromosomal composition. Most of the time it is due to hormonal influences that result in secondary sexual differentiation. And then truly the last point on this particular slide is the term unisexual dioecious or gonochoric. In this particular situation, what we are talking about is an individual that contains only male or female reproductive organs. So individuals are only male or only female, and these terms are utilized in different aspects. For example, dioecious is utilized to describe the plant kingdom and this particular phenomenon. The alternative to that is the term bisexual, monoecious, or hermaphroditic. This refers to an individual that contains both male and female reproductive organs. Again, very common in the plant kingdom. In previous lectures, you have learned about sex linkage, sex-limited, and sex-influenced traits. So again, what we'll do is transition in this lecture and talk about how that sex is actually determined based on chromosomal composition. So just to provide a little bit of a historical background and understanding of how sex determination was first determined or established, in the late 1800s we have the work of Hermann Henking. And what he noticed was that some sperm cells in certain insects contained a structure that he referred to as an X-body. This was something that was distinct in sperm cells that he didn't find in female ova or eggs. In the early 1900s, we have the work of Clarence McClung, and he studied sperm in grasshoppers and found that some sperm cells had an extra chromosome, which he then termed heterochromosome. This was a chromosome that could be distinguished based on its morphology and or behavior. And that's the term of a heterochromosome, is again one that can be distinguished based on morphology and or behavior. And then in 1906, like the slide shows, we have the work of Edmund Wilson. And Edmund Wilson studied an insect species known as Protiner. What he established when studying Protiner was that female somatic cells contained 14 chromosomes and male somatic cells contained 13 chromosomes. And this is what he quickly established as being the XXXO system of sex determination. He found that females had 2 copies of what he referred to as an X chromosome, and males only had 1 copy of the X chromosome. So the XXXO system refers to the fact that females had 2 copies of a chromosome referred to as X, and males only had 1 copy of this chromosome referred to as X. The O or the 0 basically just is a placeholder to indicate that there is no second chromosome in this particular pair, specifically in males. Wilson went on to study other insects besides grasshopper. In this particular example, what we're looking at is an insect known as Lygus. And what he noticed when studying Lygus was that both male and female somatic cells contained 14 chromosomes, which was distinct from the grasshopper example we just talked about. Females contained 2 copies of X, and males contained 1 copy of X and 1 smaller heterochromosome. And in this particular case, that smaller heterochromosome is listed or established as a Y chromosome, distinct from the X. So if we work through the same type of example we just talked about for grasshopper, we can look at 2 individuals that are mating in this example for Lygus. Females have 12 autosomes and 2 copies of X. Again, autosomes in the blue color and sex chromosomes or X chromosome in the pinkish color. We know that when females produce gametes, there's only 1 type of gamete they can produce with respect to sex chromosomes, and that is they always produce gametes that have a copy of X in it. For the male, we have 12 autosomes, 1 copy of X, and this other heterochromosome, again labeled as Y. When we look at gamete formation in males, we can see that there are again 2 distinct types of gametes that can be produced with respect to the sex chromosome. The first example is in the middle of the slide, where you have gametes that will contain the Y, and right next to that to the right is the example of gametes that would contain the X chromosome. So if a female egg was fertilized by the first example, where you have the Y chromosome in the sperm that's produced, the result would be an individual that has 12 autosomes, one copy of X, one copy of Y, and these individuals would be male. And alternatively, if that egg was fertilized by the second sperm example, the one that contains the X, you would end up with an individual that has 12 autosomes and 2 copies of X, and these would be individuals that were female. This is referred to as the XXXY system or mechanism of sex determination. It results in a 1-to-1 sex ratio as the outcome. So the first example of sex determination is the XXXO mechanism, and that simply is that individuals that have 2 copies of X develop female, individuals that have 1 copy of X develop male. In this example, we have 2 distinct sex chromosomes, the X and the Y. Individuals that have 2 copies of X develop into females, and individuals that have 1 copy of X and 1 copy of Y develop into males. So hopefully, based off of some of the basic introductory biology courses that you've had, you already have a good understanding that this mechanism of sex determination, the XXXY mechanism, is the same mechanism that is happening in humans. And so this particular slide just demonstrates that example with humans, in that with respect to sex chromosomes, males have 1 copy of X and 1 copy of Y, meaning that when they produce gametes, they will produce gametes that contain either the X or the Y with respect to just the sex chromosomes. Females have 2 copies of X, so when gametes are produced in females, females will produce only 1 type of gamete with respect to the sex chromosome, and that is going to contain the X. In this example, you can work through a Punnett square and make a prediction about what the offspring might be with respect to sex based off of the composition that we just discussed for the top part of this figure. So in humans and in just about most vertebrate species, there is a 1-to-1 sex ratio that is produced. So now I want to introduce a few more terms. The term homogametic sex refers to the sex which produces uniform gametes with respect to sex chromosomes. So in the examples that we just looked at, either with ligas or with humans, the homogametic sex is female. Females produce gametes that have uniform or are uniform with respect to the sex chromosomes. Females produce only gametes that contain the X. Contrary to that, or in contrast to that, is the heterogametic sex. And the heterogametic sex is the sex which produces gametes with unlike or dissimilar sex chromosomes. And again, with the previous examples we just looked at, that would be males with XY composition for the sex chromosomes. And again, the gametes that they would produce could either contain the X or the Y with respect to sex chromosomes. However, it turns out that the male is not always the heterogametic sex. There are plenty of other examples in the animal kingdom, such as moths, butterflies, most birds, some fish, reptiles, amphibians, where the females are the heterogametic sex. And in this situation, the nomenclature changes and is referred to as ZZZW. And this just differentiates from XXXY to indicate that it is the females that are the heterogametic sex and the males are the homogametic sex. So that designation is listed here for you. Males have 2 copies of Z and females have 1 Z and 1 W. One last point for this particular slide is that when we talk about the heterogametic sex, they are always hemizygous for the genes on X and on Y. Hemizygous just means that you have a gene present in a single copy. So when we're talking about the heterogametic sex, going back to our XXXY example, males are hemizygous with respect to the genes on X because they only have one copy of the genes on X, because they only have one X. They are also hemizygous for the genes located on Y because they only have one copy of the Y chromosome. It turns out that sex determination in humans wasn't established until about the mid-1950s when the karyotyping technique was established. So a karyotype is a chromosome complement of a cell or an individual. The most common way of preparing a karyotype is to harvest white blood cells from an individual, culture them to produce a lot more of those blood cells, treat them with a chemical known as colchicine, which arrests cells in metaphase. It prevents mitotic spindle formation. You then collect the cells, break the cells open, fix the chromosomes so they no longer move, stain them so that they're actually visible, take a picture, and then arrange the chromosomes. So in this first figure, that's what you're seeing is sort of the raw picture that would be generated. And then with someone who is trained in doing this and organizing chromosomes, they can then cut out all of those pieces and put them all together in a very organized picture like you see now. In this particular case, what you're seeing is the chromosome complement of an individual who is female, XX, and the chromosome complement of an individual who is male, XY. There are 23 pairs of autosomes in both cases, and then in the female there is 2 copies of X, and in the male there is 1 copy of X and 1 copy of Y. In most vertebrate species, and in particular in humans, there are 2 distinct types of sex chromosomes that are labeled X and Y. In this figure, what you're looking at is a comparison of the X chromosome to the Y chromosome. And the first thing that you can see is that the X chromosome is significantly larger than the Y chromosome. And there are regions within both of these chromosomes that are colored in red and are referred to as the primary pseudoautosomal region and the secondary pseudoautosomal region. It is these portions of the X and the Y that allow for these chromosomes to pair up during meiosis, specifically prophase 1 of meiosis 1 in males. Again, males would have 1 copy of X and 1 copy of Y. And as you hopefully reviewed previously, what happens during meiosis is that homologous chromosomes do in fact pair up. Because the X and the Y are not truly homologous chromosomes, they do not contain the same genetic information, these regions that are pseudoautosomal are extremely important for allowing them to pair up in males during prophase 1 of meiosis 1. So let's focus on the Y chromosome first, and then we'll talk a little bit about the X chromosome. So in this figure, what you're looking at is a depiction of the Y chromosome. What I want to do is highlight a number of characteristics or components of the Y chromosome itself. The first being the PAR, which is the pseudoautosomal region that I mentioned in the previous slide. In this particular figure, it's colored in the yellow or goldish color, and it's at the tips of the chromosome. Again, the pseudoautosomal region is the portion that allows for homologous pairing with the X chromosome in males during prophase 1 of meiosis 1. The remainder of the chromosome that's not pseudoautosomal region is listed or labeled MSY, which is the male-specific region of Y. This is the portion that is truly found in just the Y chromosome and does not have any regions homologous to the X chromosome. However, in this portion of the chromosome, there are a little more subdivisions or designations associated with the MSY. In particular, in this figure, you have what is labeled euchromatin and heterochromatin. The euchromatin would be portions where genes would be located, and heterochromatin is the portion of the chromosome where genes are typically not located overall. Depending upon the source in the literature, the Y chromosome is listed or designated as having approximately 80 genes located on the structure. That's in distinct contrast from the X chromosome, which again is a much larger chromosome and has something in the range of about 1,400 genes located on it. In particular, back to the Y chromosome, we can subdivide the euchromatic portion a little bit more into a region of that chromosome known as the SRY, which is short for sex-determining region of Y. Within the sex-determining region of Y is a specific component known as the testes determining factor. It is the testes determining factor that produces a gene product that triggers the undifferentiated gonadal tissue to form testes. Specifically, this testes determining factor, that is a subset portion of the SRY, the sex determining region of Y, is going to be the component that determines maleness. There have been a number of examples of experiments done in other systems, such as rats and mice, where this particular component is essential for the development of a male individual. So again, what this demonstrates is that the default is to produce females, and if this portion of the Y chromosome is present, the individual will develop testes and eventually become male. And again, if it's absent, the default is to produce ovaries and be female. Now, obviously, there is a whole cascade of events that needs to occur to continue the development of either male or female individuals. However, this particular point is the start of that cascade. Again, without the testes determining factor, individuals or the default is to form or develop into a female. Having this particular component allows for the development of a male. So the X chromosome is a chromosome that is very similar in function to the autosomes. There are approximately 1,400 genes located on the X chromosome, and almost all of them are unrelated to sex. This particular chromosome is very low in gene density, but because of the fact that females have 2 copies of X chromosomes, in mammalian systems, and males only have 1 copy of X chromosomes, the question soon became, do females produce twice as much of each of the gene products on X compared to males? The answer to that came from a mechanism known as dosage compensation. Dosage compensation is a genetic mechanism that regulates the level of gene products at certain locations on the X chromosome. The result of dosage compensation is that males have equal amounts of gene products from the X in comparison to females. So how exactly is dosage compensation achieved? Well, there were studies done by Barr and Bertram, and what they first noticed when looking at stained nuclei of female cells, they noticed things that they labeled as bodies in interphase. These bodies reacted with DNA-specific dyes and were then referred to as Barr bodies. A few years later, the work of Susumu Ono proposed that Barr bodies arise from one of the 2 copies of X found in females, because these Barr bodies were found in female cells and not in male cells. And this would be an explanation for how dosage compensation could be achieved. If one of the copies of X is somehow inactivated, made non-functional, then females would have 1 functional copy of X, the same as what you would find in males who only have 1 copy of X. Examples of how dosage compensation may be achieved by these Barr bodies is seen in sex chromosome abnormalities. In this particular table, what we're looking at are the normal complement in an individual who is female, 2 copies of X, and an individual who is male, 1 copy of X, 1 copy of Y. We know in this specific chromosome composition, 2 copies of X produce females and have female characteristics or traits. 1 copy of X and 1 copy of Y produce males and have male characteristics and male traits. However, there are several genetic abnormalities that have been identified that have some unusual phenotypes or some specific phenotypes associated with them. So the first example is Klinefelter syndrome. And in Klinefelter syndrome, the most common example of Klinefelter syndrome is an example of trisomy of the sex chromosomes. In this particular case, in most situations, there are 2 copies of X and 1 copy of Y. However, there have been some other extreme situations with Klinefelter syndrome having 2 copies of X, 2 copies of Y, 3 copies of X, and 1 copy of Y. But by far the most common example of Klinefelter syndrome is trisomy of the sex chromosomes, specifically 2 copies of X and 1 copy of Y. With these individuals, the characteristics are that these individuals have male traits, tend to be taller, have a little bit smaller testes, reduced facial and pubic hair, and a number of additional characteristics or phenotypes associated with this particular syndrome. Now, if we go back to what we just discussed in this particular situation, in order for an individual to develop into a male, they need to have the Y chromosome, specifically the testes determining factor portion of the Y chromosome. And so in this case, it's not a surprise that these individuals are phenotypically male. The next example is an example known as Turner syndrome. Turner syndrome is monosomy of the sex chromosomes, and in humans, this is the only monosomic situation that is tolerated, meaning individuals can in fact be born and survive with this monosomic situation. Individuals with Turner syndrome have 1 copy of X and no second sex chromosome. Because these individuals have 1 copy of X, their characteristics or their phenotype is to have female traits. The next example is something known as poly-X females or super females. These are females that have chromosomal abnormalities with a trisomic situation or multiple copies of the X. In this particular case, individuals develop with female traits. The last example of a genetic abnormality with sex chromosomes is XYY. In this case, these individuals develop into males and oftentimes do in fact have higher testosterone levels associated with this syndrome.